So first of all, we're very happy that you can join us. And Dr. Jackie Collier, I hope I'm saying that right.
Yep.
Is an associate professor at Stony Brook University School of Marine and Atmospheric Sciences. Jackie got her PhD at Stanford University back in 1994.
Dark Ages.
Yeah. Where she did research on light harvesting complexes in cyanobacteria, and focusing on the cyanobacterial acclimation and bleaching in response to nutrient deprivation. Is that fair to say?
Exactly right. Yep.
Okay. And since then, Jackie have published many articles, and now she's leading a research group at Stony Brook University, where the overarching goal is to improve our understanding of phytoplankton ecology, population dynamics, and community structure, and with an auto-ecological approach. And cyanobacteria is the main model system.
Yeah, that's changed actually. I've moved from the light side to the dark side, but I'll tell you more about that. Yeah.
I see, because this lecture is going to be on fungi, or yes,
fungus-like. Yeah.
Okay. And I had a theory about this reading the webpage that I was assigned, because in there it said that your lab research was divided into 3 parts, basically, where one was molecular genetics and physiology of cyanobacteria.
Not a lot of that going on anymore, but maybe I'll get back to it someday.
Okay.
Yeah.
And then another was comparative ecology of small planktonic picocyanobacteria, but I guess that's also in the past. And then the third one was the utilization of urea as a nitrogen source by the marine Synecoccus.
Yeah, I gotta find this old web page and get it off the web, and yeah, make people see the newer one.
Okay,
so,
but
yeah,
based on this, I was guessing that because it mentioned that this usage of urea as a nitrogen source would need some organism to break down urea. And is that where protists?
No, actually, it turns out that most phytoplankton are perfectly capable of using urea. They have their own urease. So it's a very effective and commonly used nitrogen source for phytoplankton.
Okay.
In general, I worked on it mostly in cyanobacteria, but other phytoplankton are great at using urea.
Okay.
And there's lots of urea in the water, right? Lots of heterotrophs release urea as a waste product, and so on.
Yeah. Okay, then maybe you can fill me in on the transition to heterotrophs.
Absolutely.
Thank you.
All right. All right, so shall I get rolling then?
Yeah.
Yeah, okay, great. So I have made over the last about 20 years since I got to Stony Brook in 2002, a shift from photosynthesis over toward the heterotrophic side, and at the same time from bacteria over toward eukaryotes. So I've been making this really big shift, which is probably a really bad idea career-wise. But the organisms I'm going to tell you about mostly today, the labyrinthulmycetes, really caught my interest and have kept it ever since. So let me first say I'm happy to be interrupted. If something is unclear, if you have a question, just unmute yourself and speak up. If it's something you want to save to the end, you can put it in the chat and we'll look at it at the end. But also feel free to interrupt as I go along. And let's see if I can move. Right, so introducing myself. Yeah, I did my PhD work in cyanobacterial molecular genetics at Stanford, but I always wanted to go do oceanography. My plan was just to learn the molecular biology first. And so I went and postdoced at Scripps with Brian Palaniuk. That's where I worked on urea utilization by marine Senecacoccus. I went for a few years to Rensselaer, upstate New York. My plan was to work in Lake George because you can do a lot of the same science, and actually lakes are kind of easier because the water doesn't go everywhere. You don't have to chase it around. RPI ended up being the most dysfunctional institution I ever was at. So I looked around for an alternative and ended up joining marine sciences at Stony Brook in 2002. And came with every intention of continuing to work on cyanobacteria and phytoplankton ecology, just like the description Fabian read you. But shortly after I arrived, there was an outbreak in hard clams of a disease called QPX disease. And at the time, and I think, well, I'm afraid it's no longer true, but at the time, the hard clam, Mercenaria fishery was the single largest fishery remaining in New York State. The oysters had died off long ago. The lobsters had died off about a decade before. So the hard clams were the biggest thing left, and there was an outbreak of QPX disease, which was known from Canada, from Virginia, but seemed to have skipped over New York. And QPX turns out to be a labyrinthulmycete. So my colleague, Bassam Alam, who is our resident marine disease specialist, at the time, his lab didn't have any expertise in molecular stuff of any kind. And he wanted to bring molecular tools to learn about why QPX had just showed up in New York. So he came to ask me if I would join him in writing a proposal to work on this. And my first response was, no, I'm not really interested in sick clams. But I did a little reading about Thraustocytrids and labyrinthulmycetes and learned some of the basic background. I'll tell you in just a few minutes. And then I called him back and said, I'm in, right? Looking at this as a way to get to study this group of organisms. The paper that I gave you to read, I think probably you got it, is a paper that my colleague Josh Rest and I wrote just a little over a year ago. And it was taking a look, it was basically trying to convince cell biologists who mainly study a few model organisms, right? They study human cells in culture, they study yeast, they study Drosophila, they study nematodes, just a very few model organisms, trying to convince them that they should start working with people like us to do cell biology on marine protists, right? For all the reasons, so I don't have to tell you how important protists are in the oceans, but we have to tell that to the cell biologists. They don't have any idea. They also mostly have no idea how diverse eukaryotes really are. They don't realize that those model organisms they study, even if you include yeast, represent a tiny fraction of eukaryotic diversity, and all the rest of it is mostly in the ocean. Great taxonomic, ecological, morphological, life history variation from which they could learn a tremendous amount about where eukaryotes came from, what our evolutionary potential is, how all of our cell biology works, etc. And so we were pushing that idea in this paper that none of these organisms have yet achieved that status of model organism. And we gave a few examples. In that paper, we presented a tree showing some drawings and emphasizing some in green photosynthetic organisms that could potentially become models, some of the heterotrophs that are the closest unicellular relatives of animals. and emphasizing a few examples of some of the things we could learn from studying marine protists. And I'll be happy to come back and talk about anything to do with this paper later, or to answer any questions anyone has right now. But otherwise, what I wanted to do is actually talk about my favorite organisms rather than marine protists in general. And my favorites, of course, they got to star in this tree. They got much better representation in this tree than most of the other eukaryotes because I made the tree. So there are 5 different representatives of labyrinthulmycetes in this tree. Arantiochytrium, I'm going to tell you a lot about today, Schizochytrium, aplano, Labyrinthula, and oblongi. So let me tell you a little about these organisms and why I think they're so interesting ecologically, cell biologically, all kinds of other reasons. Okay, so brief introduction. This is a microscope image. The big brown spheres are pollen grains that my student in ICSI dropped into a sediment slurry from Peconic Bay, which is just on the east end of Long Island. She left the pollen grains in there for a couple of days and then took them out and took this photograph. And you see these clear cells. Let me see what I can draw with. You see these clear sort of round cells attached to the outside of the pollen grains. These are the vegetative cells or the thalli of labyrinthulmycetes. What happens is if you drop pollen grains in seawater or a sediment slurry from pretty much anywhere in the ocean, zoospores from cells that were already in the culture are going to find their way to the pollen grains, settle, attach themselves, and start growing. And this is actually the most common way by far to isolate labyrinthulmycetes from the ocean, because they are just about the only thing in the ocean that can grow on pollen grains. Pollen is really famous for how refractory the wall is. And so labyrinthulmycetes are kind of unique in the capacity to do this. Of course, they can't all do this, so that has skewed our understanding of their diversity, but it's also enabled what understanding we have. Okay, where's my little thing? Okay, I put this early so I would remember. So this is, get rid of my drawings. Just an introduction to some of the people in my lab who have worked on labyrinthulmycetes over the years. My technician, Joanne, my student, Nixie, whose image I just showed you. Chin Chen was the first to work on QPX. My colleague, Glenn, and Ari was a high school student who worked with us on some data I may or may not have time to tell you about later. And right now, I'm working again with my colleague, Josh Rest. My graduate student, Mariana Rios, is getting very close to defending her dissertation. And our postdoc, Anbu, I'm going to show you some of his work shortly. Our work these days has been funded by the Moore Foundation as part of their program to do exactly what I wanted to do, which was develop molecular genetic techniques to work on more marine protists. So it was perfect opportunity for us. Okay, labyrinthulmycetes are ubiquitous marine organisms. Even if you've never heard about them before, that doesn't mean they're not out there. They are in sediments everywhere from the tropics to polar regions, including in the deep ocean. They're in the water column, again, all over the oceans from the surface to great depths. And they can be quite abundant. Abundance estimates, which are mostly based on that pollen baiting method or an MPN, a most probable number method using pollen baiting, range from 1,000 to 1,000,000 cells in a liter of seawater, 100 to 10,000 in a gram of sediment. And those might be undercounts because of the biases in those methods. So they're everywhere, they're abundant, they show up in all the high throughput 18S sequencing efforts. They're not the most abundant thing in there, but they're in there. Raghukumar, Seshagiri Raghukumar is maybe the best known person to work on labyrinthulmycetes. His review papers from '92 and the early 2000s are still a couple of the best references on the ecology of these organisms. And he referred to them as the most common fungus-like organisms in the sea. They are not funguses, but their ecology is fungus-like in that we think they mainly live as saprobes. They break down dead particulate organic matter. Plants, algae, animals. As I just showed you, some of them can degrade really refractory materials. Others apparently cannot. They also appear to be really widespread as commensals and sometimes as parasites of marine plants and animals. So for example, QPX disease in hard clams. We find QPX disease in clams all the time. Now that we have good methods with which to detect it, we find that QPX is basically always in hard clams, even healthy clams. Particularly, it seems to be associated with the paleo cavity. So it kind of lives maybe inside the shell, closely associated under normal conditions. And we think that it's an opportunistic parasite. So it's a commensal, it always lives with hard clams. And when something goes wrong, the clam gets sick or something else, then QPX disease develops as QPX invades the host tissue. Another example you might have heard of is eelgrass wasting disease. Labyrinthula is the genus from which the whole group takes its name, and I'll tell you a little more about that in a bit, is associated with eelgrass wasting disease. And the story there appears to be similar. Labyrinthula appears to be a common commensal of seagrasses, but under some conditions becomes pathogenic and causes a disease known as eelgrass wasting disease, can take out whole eelgrass meadows. And there are thought to be other examples again as well. One of the nifty stories that's kind of hiding in a paper that's difficult to find is the note that you don't see a lot of work on cell cultures from marine invertebrates, right? You see a lot of cell culture work from other animals, not so much from marine invertebrates. And apparently one of the reasons for that is the cultures often get overtaken by labyrinthulmycetes, suggesting that marine invertebrates are commonly very closely associated with these organisms, and we know basically nothing about that. Ecologically, a really interesting aspect of labyrinthulmycetes is that they can synthesize de novo and store really large amounts of essential omega-3 long-chain polyunsaturated fatty acids, like DHA. In fact, you could go to the health food store now and you could buy an omega-3 supplement instead of fish oil that's made from labyrinthulmycete. On the label, it'll probably say schizachytrium. It's not actually schizachytrium, that's a whole taxonomic mess, but there are multiple companies out there using these organisms to produce PUFAs. That's where the supplements that go in baby formula come from. That's where supplements that are going in a variety of foods and fish feed and other things are coming from, to try to replace the incredibly high demand for these same fatty acids that's being put on wild fish populations to sustain that need. And in fact, a lot of what we know about the genomics, the biochemistry of these organisms is because of this biotech interest. I think I already said most of this. Up to half the biomass can be lipids in particular strains grown under the right conditions. And the people using these strains to produce PUFAs are interested in using them to produce other things as well. Potentially carotenoids, which I'll tell you more about, squalene, extracellular polysaccharides, perhaps other things as well. So this should also tell you that at least some of these organisms are super easy to grow in the lab, and grow them in basically the same media that you would grow bacteria. Would you grow E. coli? So little glucose, little yeast extract, little peptone, little instant ocean, and many of them are perfectly happy. They are obligately marine, you have to add salt. But otherwise, pretty standard. And some of them can grow really fast to really high biomass. Others, not so much. All right, so here's a different tree of eukaryotes. It's maybe a little easier to see here where the labyrinthulmycetes fall with my big red arrow. So what I want to point out here is that the fungi are way over here. Fungi are opisthecons. They're on the same part of the tree that we are. Here's all the animals on this little tiny branch over here. The labyrinthulmycetes are completely on the other side of the tree. They are a basal branching lineage in the large group known as the straminophiles. And you are familiar with straminophiles whether you know it or not, because the photosynthetic crown group of straminophiles includes diatoms, kelps, a lot of other photosynthetic eukaryotes you're familiar with. The other groups you might be familiar with, all these deeper branches are heterotrophic. It's only the crown group that's photosynthetic. The oomycetes may not be familiar to you from that word, but if I say Irish potato blight, that's an oomycete. In fact, a lot of plant pathogens are oomycetes. The bicosoecids include heterotrophic nanoflagellates, major grazers in marine food webs. These guys back here, the opalinids, are kind of weird gut commensal bacteria that I don't know much else about. Yeah, I think those were my main points for the taxonomy. And again, the point for most people, cell biologists, large organism ecologists, evolutionary biologists, is they think this is eukaryotes, right? And I like to try to convince them that that's hardly anything. This is what eukaryotes are capable of. Okay, so let's zoom in on the labyrinthulmycete branch. There's 3 major groups known from cultivated strains. The labyrinthulids or labyrinthula, whoops, sorry about that, which are the ones associated with eelgrass wasting disease. A planochytrium, which actually seems to be the most abundant type in the oceans, especially in coastal settings like around Long Island. Unfortunately, they are not super easy to grow in the lab, and we don't yet know very much about them. Oblongichytrium, which for a long time were mistaken as being thraustochytrids and only recently are recognized as a completely different group. And then this very big group here, the thraustochytrids. These groups are all monogeneric at this point. There's lots of named genera in the thraustochytrids. There is really high diversity, even though the taxonomy is a complete disaster. You really can't trust what most of these organisms are called, but there's lots of diversity in this group. To give you some idea, a little more idea what they look like. So this is a sketch of what would have been that clear glob of vegetative cell you would have seen attached to the pollen grain. This is a structure called the Ectoplasmic Network, by which it attaches to the pollen grain. And here's a little sketch of a zoospore. Now, this looks like a sporangium that's actually about to release a bunch of zoospores. The planochytrids are distinct from the thraustochytrids in that they can actually move around on their ectoplasmic network. And Daisuke Honda has shown that they can also use their ectoplasmic network to grab diatoms, bring them in and digest them. So this is maybe a sort of predatory purpose in aplainochytrids. The labyrinthulids are actually colonial. They have an elaborated ectoplasmic network that's shared by a bunch of cells, and they move around inside that. And they form colonies associated with seagrasses in that way. The vegetative cells are typically 5 or 10 microns in diameter, something along that line. The diversity I mentioned. So this is a tree just of the Thraustochytrids. And I want to make the point that the 18S ribosomal RNA sequences between pretty much any 2 genera of Thraustochytrids are as different from each other as you are from a nematode or a sponge or a ctenophore, right? So there's animal scale diversity just within the Thraustochytrids. Okay, I decided I would put this here a little bit more on the typical life cycle. So vegetative cell that eventually forms zoospores, which I gotta quit doing that, form more vegetative cells. That is a common trait shared by all the genera. You'll notice sporangium release zoospores, get a new vegetative cell. Sometimes I'll show you a video of this amoeboid cell thing that they do that we don't know anything about, that I would love to know more about. But the main dispersal stage is the zoospore. This is zooming out. So this is the Thraustocytrids I just showed you earlier. Here's the Labyrinthula, Aplanos, and Oblonges. And you'll notice there's a whole bunch of other groups in here, which we only know from environmental sequences. So there are a lot more diversity of labyrinthulmycetes in the ocean than we've cultivated yet. We haven't been able to cultivate any of these other groups. And actually right now, I'm gonna say it remains really unclear exactly where the limits of labyrinthulmycete are based on all of these sequences. I tend to be really conservative about which environmental sequences I call labyrinthulmycetes. Other people are taking a much more generous approach and calling almost anything that doesn't clearly go somewhere else a labyrinthulmycete. So it's going to be a lot to resolve there. Okay, let's see. I'll tell you just a couple of the cell biology things we're interested in, and then see what you want to hear about, right? So in terms of cultivated labyrinthulmycetes, the feature that unites them as a group is a structure associated with that ectoplasmic network that's known as the bothrosome. This is an electron micrograph image. And this structure here connects the ectoplasmic network, which is branching extensions of the cytoplasmic membrane. It comes in here and it gets connected to the endoplasmic reticulum through this bothrosome. Nobody has any idea at a molecular genetic level what that's made of, or in an evolutionary sense where it came from and what it's related to. And those are questions we would really like to know the answers to.
Excuse me.
Yes.
Can you take that again?
Sorry?
Can you take that again?
Go through this again?
Yeah, the bothrosome.
The bothrosome, okay. Oops, uh-oh, I can't if I can get back to it. Okay, there we are. So I showed you, let's go back. So this structure, this ectoplasmic network that attaches the cell to the surface, the point where it comes into the cell body, this thing is unique. It's not like any other cytoplasmic extension that's known among any eukaryotes. So this is a unique bit of cell biology to do with labyrinthulmycetes. The other thing that's unique is the organelle that's found where the ectoplasmic network comes into the cell body. So you can't read the ectoplasmic network in this, it's coming of the cell body in a different plane. But this structure is where it comes in and connects to the endoplasmic reticulum, the endoplasmic membrane system of the cell, where a lot of the basic biology happens. So in theory, what's going on here is that digestive enzymes, for example, are being produced, excreted out into the ectoplasmic network. It's digesting the substrate it's living on. Digestive products are being taken back up in the ectoplasmic network and returned to the cell body through that bothrosome. At least that's what people think is going on. Did that make better sense that time?
Yes, thank you.
Okay, great. Any other questions on that? Yeah, so that's something we really, really want to know about. So I'm gonna skip over that one. So this movie is showing over and over again a zoospore escaping and swimming off. You can also see some other zoospores, like, oh, it stops the movie when I do that. Okay, you'll see this zoospore, here's another one that are kind of stuck. Sometimes you can even see the flagella wiggling. Zoospores tend to get stuck to slides by their flagella. So the reason I'm showing you a zoospore is because the ectoplasmic network forms when the zoospore settles and attaches to a substrate. And this is a several hours time-lapse image of some newly settled zoospores. And if we wait just a little bit here, you're going to start to see the ectoplasmic network extending out from the cells. You can see these thread-like extensions attaching to the glass slide and searching their surroundings for food, basically. So this is what Anbu's been working on. One of the things he's working on. Now that we sort of have the timing down, we're hoping to get our hands on some money so we can do some proteomics and transcriptomics to look at what proteins are being produced while this is happening, to help us get our hands on what's the bothrosome, what genes are involved in that, how's it being made. So that's where we're at on working on the bothrosome. I promised I would show you a movie of the limaciform or slug-like cells. This is another video with about 6 hours compressed to 40 seconds, and here they come. So these slug-like cells crawl around, and then they stop, they settle down, they round back up, and they start growing. The strain we work with, Aurantiochytrium limacinum, gets its species name because it tends to do this a lot. But a lot of other Thraustochytrids seem to do this as well. And we don't know anything about the cell biology of this process. You can watch it happen again because it's so cool. If you're actually watching this in real time, it's not super obvious. Only in the video do the amoebae look quite so active, but they're not amoebae at all in the typical sense of an amoeba in the way they move. It's just kind of, for lack of a better word, that we call them that.
Jackie, can you trigger this, or it just?
Yeah, although I will admit that we've tried this a few times and we certainly don't understand exactly what the triggers are. Right? How they decide, now I'm gonna make limaciform cells versus now I'm gonna make zoospores, to me is one of the really most interesting questions. Yeah. And those are two really different dispersal modes, right? So probably something about the environment gives them a cue about whether I want to disperse far away, therefore I make zoospores, or whether maybe I don't want to disperse so far away, so maybe I make limaciform cells. I hypothesis that I would love to have a student come and figure out how to test that. Yeah. Well, we need to get our hands on some of these genes so we can make mutants, and then really do experiments like that, right? The other cell biology feature we're working on, I mentioned biotech interest in carotenoid production by Aurantiochytrium. And this is an auger plate with colonies of Aurantiochytrium growing on it. And you can see how orange they are. It is not always orange. It depends on the media composition. And for some reason we cannot explain, it also depends on light. So on richer medium and in the light, and as cultures get old, you can get really striking orange color like this. And they're accumulating mostly astaxanthin, mostly a xanthophyll. And this is a really unusual thing for a heterotroph to do, right? Carotenoids are absolutely essential in photosynthetic organisms. You cannot do photosynthesis without carotenoids for protection from overexcited chlorophyll, from oxygen radicals, etc. Why a heterotroph needs to have carotenoids is a little bit mysterious, and not very many heterotrophic eukaryotes do. There are a couple of fungi. There are actually a couple of animals. Plant-associated mites have actually gotten these genes by horizontal gene transfer, and they make their own carotenoids. And there's Strauss-Tolkitrids. And so it's mysterious. It's really interesting. I think it probably contains a useful clue about what their life is really like in the ocean. They don't all make carotenoids. Some make carotenoids, and the ones that do only make them under particular conditions. So there's something we can learn here about their ecology. We have a hypothesis about why they make carotenoids, and our hypothesis is that it's related to that storage of huge amounts of fatty acids. So these are DIC and fluorescence images of the same group of Aurantiochytrium cells that have been stained with a dye called Nile red. Nile red fluoresces this really lovely gold color when it's in the appropriate hydrophobic conditions, like what you get in a lipid droplet. And that's how labyrinthulmycetes store their lipids, is in big lipid droplets. So our hypothesis is that the carotenoids are also in the lipid droplets to protect the fatty acids from oxidative damage, because fatty acids are really sensitive to oxidative damage. So how do we test that hypothesis? In the genome, it was actually pretty easy to identify a gene that looked like it was involved in carotenoid biosynthesis. And this is almost entirely unique to the Strauss-Tolkitrids, although we found it also in Thecamonas, which makes no sense to us evolutionarily, but there it is. This one gene has fused together phytoene synthase, phytoene desaturase, and lycopene cyclase, which are the three activities that are required to convert geranyl diphosphate to beta-carotene. So this is how they make carotenoids. What Mariana has done is to knock that gene out. And I hope that maybe you can see, this is the wild type. The pellet's a little bit orange there. This is the knockout. It never turns orange. We did indeed stop it from making carotenoids. This is an absorption spectrum of extracted carotenoids from the wild type in rich media where it makes lots of carotenoids, and a poor medium up here where it only makes a little bit of carotenoids. And this is the mutant where it doesn't make any carotenoids. So Mariana has knocked out carotenoid production and is trying to right now get the experiments going to test our hypothesis about this. And I think I will stop there and answer questions. And I'll tell you more stuff if we run out of questions, but for now, I'd like to hear your questions.
I have a quick question just about this.